Stem cell therapy stands as one of the most compelling possibilities in regenerative medicine, and its potential to reverse cellular aging through telomere restoration is drawing increasing scientific attention. At the heart of this approach is the ability of stem cells, particularly embryonic stem cells and induced pluripotent stem cells, iPSCs, to maintain long telomeres and high telomerase activity, enabling them to divide repeatedly without entering senescence. By transplanting these telomere-rich cells into aging or damaged tissues, researchers hope to replenish worn-out cell populations, promote tissue repair, and potentially reset the biological aging clock at the cellular level. The biological foundation of this approach is strong. Telomeres, the protective caps at the ends of chromosomes, shorten each time a cell divides. Once they reach a critically short length, cells enter a state of growth arrest known as replicative senescence. Stem cells, however, are an exception to this rule. They express telomerase, the enzyme responsible for adding telomeric repeats, and can thus preserve their chromosomal stability and regenerative potential for far longer than most somatic cells. This characteristic makes them ideal candidates for cell replacement therapies aimed at rejuvenating aged organs, whether in the heart, brain, skin, or immune system. Transplanting young, healthy stem cells into aged tissues could, in theory, create a local environment in which new, functionally youthful cells outcompete the senescent ones, leading to improved tissue function. For example, in preclinical studies involving mice, mesenchymal stem cells, MSCs, or hematopoietic stem cells with long telomeres have been shown to enhance repair processes, reduce inflammation, and improve longevity markers in aged recipients. In humans, early clinical trials using autologous or donor-derived stem cells for heart failure, neurodegeneration, and immune rejuvenation have shown promising, albeit variable, results. Some of these effects may stem not just from the cells themselves, but from their ability to secrete growth factors and anti-inflammatory signals, sometimes referred to as a paracrine rejuvenation effect. A particularly exciting avenue involves the generation of patient-specific iPSCs. These are somatic cells that have been reprogrammed back to a pluripotent state and, in the process, have their telomeres reset to youthful lengths. When differentiated into tissue-specific cell types, iPSCs can in theory replace damaged cells without triggering immune rejection, since they originate from the patient's own tissues. Furthermore, because iPSC reprogramming reactivates telomerase and repairs mitochondrial and epigenetic damage, they may offer a deeper form of cellular rejuvenation than conventional cell transplants. Despite the promise, there are real and complex challenges to overcome. One major concern is the integration of stem cells into host tissues. For stem cell therapy to be effective, transplanted cells must not only survive but also differentiate correctly, form proper connections, and function in harmony with native cells. This is particularly difficult in complex organs like the brain or heart, where cellular architecture and signaling are highly specialized. Misplaced or improperly differentiated cells could disrupt tissue function or trigger immune reactions. Immune rejection remains another formidable obstacle, especially when using donor-derived stem cells. Even with close genetic matching, minor histocompatibility differences can provoke an immune response, limiting the longevity and effectiveness of the transplant. Immunosuppressive drugs can mitigate this, but long-term use carries risks. Autologous iPSC-derived therapies may eventually solve this problem, but these approaches are still expensive, technically demanding, and not yet widely available outside of research settings. There are also concerns about the long-term safety of stem cell-based interventions. Because stem cells possess the ability to divide indefinitely, they carry a theoretical risk of forming tumors or contributing to unchecked cell growth if not carefully regulated. The telomerase activity that makes them rejuvenating can, under the wrong circumstances, make them prone to transformation. Thus, rigorous screening, safety controls, and precision delivery systems are essential before these therapies can be safely scaled for routine use. Still, the long-term vision is compelling. By combining the regenerative capabilities of stem cells with advances in gene editing, bioengineered scaffolds, and targeted delivery, it may one day be possible to rejuvenate organs not just structurally, but at the molecular level. Rather than merely slowing aging, Stem cell therapies could offer ways to reverse its effects, replacing senescent cells, restoring telomeres, and reawakening youthful function in aged tissues. In the current landscape of longevity science, stem cell therapy remains a beacon of hope. While challenges around safety, cost, and immune compatibility remain, the underlying science continues to mature. As protocols become more refined and supportive technologies evolve, we may soon see stem cell therapy move from an experimental promise to a cornerstone of age reversal medicine. With each step forward, we get closer to the extraordinary goal of re-engineering aging at its root, the cell itself.